Dr. Wilkins is an active member of Heartland Center for Mitochondrial Medicine, in which I'm a co-founder of this organization. She successfully organized 2 meetings, and I am very thankful for her services. Dr. Wilkins presents 29 publications in reputable journals. She further co-authored the 10 book chapters and editorial comments. Her works were cited 1,498 times according to Google Scholar with the H-index 20. It's a pretty impressive for a young scientist.
You're telling me things I didn't even know about myself. Thank you.
Dr. Wilkins is currently holding the K1990A/R00 grant. It's a training grant for young investigators to become an independent investigator. She had received multiple small intramural pilot grants too. You know, Dr. Wilkins and I have common research interest. We are both mitochondric, and we love to study mitochondrial biology. Without further ado, please join me to welcome Dr. Wilkins. Dr. Wilkins?
Well, thank you, Dr. Agbas, and thank you so much for having me today for this great opportunity to speak about the research that I am currently in charge of now as a young PI. So I know you mentioned biomarkers. I do want to say I'm not going to talk much about biomarkers today, but I am actually now the Associate Director of our Biomarker Core for the KU Alzheimer's Disease Center. It's a new position, so we're still figuring out what that means. But I will be in charge of novel biomarker development, which means that I will be trying to figure out what types of mitochondrial biomarkers we might be able to use in neurodegenerative diseases, not only to look at, you know, how maybe a patient is progressing in their disease, but also if a drug that we're testing may be affecting mitochondrial function. And this is a challenge because, as you know, in neurodegenerative diseases, we cannot, you know, take samples of somebody's brain. So we typically have to use blood, muscle, or skin for that. And so that's an ongoing project. If anybody has any questions, I would be welcome to answer them at the end of my talk or during my talk. Just let me know. So today I'm going to speak about my K99R00 project, which looks at amyloid and bioenergetics. So let's first though talk about why amyloid and Alzheimer's disease. So I know probably many of you have heard or are familiar with, and hopefully not, but unfortunately know someone who's affected by Alzheimer's disease. It is the most common form of dementia in throughout the world. And just so you're aware, there are many other types of dementia. Some of them that I actually was not aware of until I moved here and studied under Dr. Swerdlow. But in Alzheimer's disease, one of the first findings by Louis Alzheimer in 1906, so the early 1900s, was if you look at the brain of someone at autopsy, then we see amyloid plaques, which are shown here, and tau tangles. And the tau tangles are a protein, so it's a protein called tau, and it becomes hyperphosphorylated, and it creates intracellular tangles in neurons. And the amyloid is derived from amyloid precursor protein, and I will go through how its cleavage process in a little while, but it forms extracellular plaques. And so you can see these plaques and tangles throughout the brain in someone who has Alzheimer's disease. And this currently is the definition of Alzheimer's disease in the field. So Alzheimer's disease is not definitively diagnosed until autopsy. We also, more recently in the past decade or so, have been fortunate to have some very intelligent people develop radio-labeled tracers, which can be used in PET imaging or positron emission tomography imaging, to where we can actually see amyloid in the brain of a living subject. So this is PIB, this is the red and the yellow are showing high levels of amyloid versus, blues, which are low or negative control, in the cognitively normal subject here. This PBB3 is actually showing phosphorylated tau. So this will show up the tau tangles in the brain, again, with red and yellow being high levels, green and blue being little to no levels. And this is another amyloid scan with a different compound called florbetabar, which we actually do use in our center. And you can see someone who's cognitively normal, they may have a little bit showing up, but someone with Alzheimer's disease, they obviously have a lot more based on the red and yellow color. So overall in Alzheimer's disease, there are protein aggregates forming in the brain. So there are a few different hypotheses of what causes Alzheimer's disease, and I'm going to go through those because they're important to understand why we are interested in our current research track. The first hypothesis is called the amyloid cascade hypothesis, and this hypothesis places amyloid as the first triggering event that induces neurodegeneration and cognitive decline and dementia. And so this hypothesis is actually based on what we call early onset familial Alzheimer's disease subjects. The genetic mutations that they have are usually in amyloid precursor proteins, so that's where amyloid beta is generated from, or presenilin 1 or presenilin 2. And these are enzymes which are a part of a complex that generate amyloid beta. So mutations in these genes in humans does increase amyloid beta production. And because there's more amyloid beta, it's believed that this increases amyloid beta monomers, and then eventually the oligomerization of those monomers, so building up into larger protein complexes that then can lead to plaque deposition. And that these plaques are what cause synaptic and cellular changes, including loss of neurotransmitter signaling, oxidative stress, inflammation, mitochondrial dysfunction, and eventually cell death of neurons. So, this hypothesis seems pretty relevant, especially to familial Alzheimer's disease subjects who have these mutations. But it's important to note that less than 1% of the population of Alzheimer's disease subjects have familial mutations. The majority of Alzheimer's disease subjects are actually of sporadic nature, meaning that they are late onset, so age 65 and older, and there's no known genetic mutation directly causing their Alzheimer's disease. There are genetic variations, though, that associate with late onset Alzheimer's disease. One of them is apolipoprotein E, or ApoE, and there are 3 alleles, 3 different single nucleotide polymorphisms for ApoE. One of them is the most common, and that has a neutral risk. One of them is a little less common, and it may decrease your risk for Alzheimer's disease. But the other one, called ApoE4, is about 10% of the population, and it increases your risk, and it's in a dose response. So if you have 1 allele, it increases the risk about 2-fold, but if you have 2 alleles, then it increases your risk about 5 to 6-fold. And what's interesting is that in our Alzheimer's disease population, while ApoE4 is only about 10% in the general population, ApoE4 accounts for close to 60 to 70% of our Alzheimer's disease subjects. So it is there is a higher amount of ApoE4 alleles in our Alzheimer's disease cohorts. So ApoE has been linked not only to lipid metabolism, but also amyloid beta clearance. And so it's believed that these genetic factors will reduce amyloid beta clearance, which then increases the amount of monomers, and again, leads to these downstream effects. So either way you look at it, the amyloid cascade hypothesis is stating that amyloid beta is what causes Alzheimer's disease. The other hypothesis is places tau upstream and states that tau fibrils and oligomerization cause Alzheimer's disease. Whether it be that there's a genetic mutation in tau, there are some genetic mutations in tau, and more recently, possibly epigenetic factors that could affect the phosphorylation of tau. And that once tau becomes hyperphosphorylated, you lose microtubule stability, and this really affects neuronal function and their ability to actually transport cargo up and down the axon, which is really important for synaptic signaling. So this loss of microtubule stability and this tau hyperphosphorylation can lead to neurofibrillary tangles and eventually neuronal degeneration and cell death. So this hypothesis places tau above amyloid, and it also, in some of the iterations of this hypothesis, states that tau can lead to amyloid beta oligomerization and plaque formation. Whereas some of the amyloid cascade hypothesis state that amyloid beta plaque formation can lead to tau hyperphosphorylation and tau tangles. And so basically what we're kind of fighting here in these hypotheses is what came first, the chicken or the egg. Well, Dr. Swerdlow, who I completed my postdoctoral fellowship under, he proposed a couple decades ago the mitochondrial cascade hypothesis of Alzheimer's disease. And this hypothesis places mitochondria above everything else. So that mitochondrial dysfunction is actually upstream of all other pathologies that we see in Alzheimer's disease. And this hypothesis states that your baseline inheritance of your mitochondrial function and density will really determine your risk for Alzheimer's disease, because everyone's mitochondrial function declines with age. And there's some threshold that we each have, and we all have different thresholds for what this is. And this can be possibly changed through lifestyle factors like exercise and diet. But once this functional threshold is released, and you get overt mitochondrial dysfunction, that's what leads to tau hyperphosphorylation and tangles, amyloid beta plaque formation, and then synaptic loss and neurodegeneration, which ultimately culminates in dementia. And so again, you have an amyloid cascade hypothesis, a tau hypothesis, or a mitochondrial cascade hypothesis, each placing one event above the other in what is actually causing the pathologies that we're seeing. So I think it's really important to talk about how amyloid beta is made, since I'm going to be talking about relationships between amyloid beta and mitochondria today. So amyloid beta is actually generated from amyloid precursor protein. This green molecule is representing that protein. It is a transmembrane domain protein, so generally it's found in plasma membrane or embedded in membranes. As I will speak to later, it is actually found in mitochondria and mitochondrial associated membranes as well. There are many different isoforms of amyloid precursor protein, so there are different lengths of the protein. It is ubiquitously expressed in most tissues of the body. In the brain, though, it's expressed as a 695 amino acid protein. In other parts of the body, it's expressed as like 770 or 717 amino acids. It can vary in size. So it's a transmembrane protein. It really likes to be stuck in lipids. It's not very soluble. And it can be cleaved in two different pathways. So depending upon which enzyme cleaves first will determine if we generate amyloid beta or if we do not. So if alpha secretase cleaves amyloid precursor protein first, we generate soluble APP alpha and a small C-terminal fragment known as C83. And this C-terminal fragment is then cleaved by gamma secretase. Gamma secretase is a very large enzyme that's composed of 5 to 6 different proteins. And this is where those presenilin mutations come into play, because presenilins make up the kind of the catalytic core of this enzyme. And so this C-terminal fragment is cleaved by gamma secretase to generate the small P3 fragment, which no one has any idea what the function of this is, or the APP intracellular domain. Now, what's interesting is the APP intracellular domain does localize to the nucleus with a cofactor, Fe65, and it can function to change gene expression and transcriptional activation. Soluble APP alpha is thought to be a neurotrophic factor, meaning that it might be important for synaptic health and function in the brain for neurons. And some people pose that loss of soluble APP alpha may be what drives Alzheimer's disease and not actually amyloid beta production. So if we actually cleave with beta secretase instead of alpha secretase first, then we generate soluble APP beta, which is missing a large chunk of amino acids found here. And so this does not have neurotrophic properties like soluble APP alpha. We generate a larger C-terminal fragment known as C99. And when gamma secretase cleaves this fragment, we generate amyloid beta, again, an APP intracellular domain. What's interesting is that this intracellular domain is 2 amino acids shorter than this one. And so there may be functional differences here as well. It's also important to note that gamma secretase does not just cleave these C-terminal fragments one time. It actually does a trimming function, kind of like a proofreading. So it will actually cleave 3 separate times and generate different fragments of amyloid beta. And so you can get varying species sizes of amyloid beta, ranging from 37 amino acids to 43. And there are some people who think that you can also get even longer forms of amyloid beta, such as those that are 48 and 49 amino acids long. The length may be important because that could dictate its ability to form plaques and how kind of sticky the protein gets. Eventually, when you get enough of a protein, it's probably going to fall out of solution. It's kind of like if you continue to just keep pouring salt or sugar into water, eventually you'll reach a point where it's no longer soluble. And a lot of people do focus on amyloid beta 42 because in particular, this one, the species has been known to be more prone to aggregating, so less soluble. And amyloid beta 40 and 42 ratios are typically a biomarker that are looked at in Alzheimer's disease. I think it's also important to talk about the evolution of the amyloid hypothesis, just so you can understand some of the confusion that's still ongoing in the field. And it's important to keep in mind that amyloid beta, when it's first generated, is a monomer. And when those monomers come together, they will form oligomers. Those oligomers can form then into protofibrils, and those protofibrils will turn into fibrils. And ultimately, this is So as I said, in 1906, Alzheimer's disease was discovered by Louis Alzheimer. And then fast forward into the 80s, there wasn't a lot that happened in this time frame. And you can actually find this information at the Alzheimer's Association website as well. They have an interactive tool where you can look at literature from each of these time points, and it will take you to the original research paper, which is actually really cool. In 1984, amyloid beta was identified as causing these plaques that Louis Alzheimer was seeing in the brain. And then 5 years later, it was found that amyloid beta is toxic in vitro. So in cell cultures, if you put amyloid beta onto cell cultures, it does cause the neurons to die. In 1990, alpha secretase was discovered. So that's the enzyme that if it cleaves APP first, you do not generate amyloid beta. In 1995, they decided that the oligomers were toxic, not just the soluble form of amyloid beta. So that's right here. But then 2 years later, it was switched to protofibrils. And then just a couple more years later, they decided again that soluble amyloid beta was the toxic species. And presenilin was discovered. And it was actually discovered by Dr. Michael Wolf, who is at KU Lawrence currently. He moved here from Harvard. And he was the one who really showed that presenilin was the catalytic subunit of gamma secretase. In 2000, we finally were able to get a structure of base, and base is beta secretase. So that's the enzyme that if it cleaves first, amyloid beta will be generated. But then in 2001, we switched over to saying fibrils were toxic. But then a year later, we were saying oligomers were toxic, getting a bit of whiplash now. In 2003, this was really interesting because it showed that activity-dependent amyloid beta release and synaptic depression was important, so that amyloid beta was released from neurons during synaptic activity, which is really interesting. In 2008, apolipoprotein E was discovered, and amyloid beta clearance was discussed by ApoE. Actually, ApoE wasn't discovered, but it was discovered that ApoE was involved in amyloid beta clearance. I apologize for that. And then in 2010, ADAM10 was identified as the alpha secretase enzyme, this protein. And in 2013, I think one of the most important discoveries so far was found based on the advent of neuroimaging for amyloid beta and tau, and that there's actually a 20-year prodrome to Alzheimer's disease, meaning that amyloid beta accumulates in the brain decades before dementia onset. And so this is actually really important and has structured a lot of our ongoing research. So as I said, when someone's cognitively normal, they're actually accumulating amyloid beta in their brain to an abnormal point. And it seems to be that when amyloid beta is no longer being accumulated in the brain, that's when dementia onset starts. And I think this is actually really important. It's also important to note that if we're talking about the different hypotheses, tau is actually the only biomarker that positively correlates with dementia. Amyloid beta does not correlate well with dementia or cognitive ability, but tau does. And so that's actually very interesting. So here comes in where I'm going to talk now more about my favorite subject in the world, which is mitochondria. In Alzheimer's disease, mitochondrial dysfunction is noted. It's very well accepted. There is decreased cytochrome oxidase expression in neurons. So cytochrome oxidase is complex 4 of the electron transport chain, and it's the enzyme that consumes oxygen and hands off electrons to complex 5, where ATP is generated. It's a very important enzyme, especially for maintaining energy production. We also see decreased cytochrome oxidase Vmax. So what that means in the enzyme world is that the enzyme is not built the same way in someone who has Alzheimer's disease. There's something structurally different about that enzyme. It's not functioning at the same level. And we use that as a biomarker in some of our clinical trials. We can measure these changes not only in the brain, we can also measure them in skin and blood cells. So this is not just a brain-specific mitochondrial defect, it's systemic. We also see increased mitochondrial DNA mutations and deletions in the brain of subjects who have Alzheimer's disease versus someone who does not. We see decreased brain glucose utilization and metabolism. This was actually one of the first findings in the 1980s as well, independent of amyloid. The brain is either not taking up glucose or it's not utilizing it to the same extent as someone who does not have dementia, showing an overall metabolic defect in the brain. So as I mentioned before, APP and amyloid beta are known to localize the mitochondria. Some people still kind of argue about this, but based on my own data, I'm pretty comfortable saying that amyloid precursor protein does localize the mitochondria. And so what's actually interesting is that there might be several different ways that it localizes the mitochondria, in that it may actually insert itself into the translocases of the outer mitochondrial membrane and the inner mitochondrial membrane, and it could get stuck in these ports, preventing other proteins from getting imported into the mitochondria, which could affect mitochondrial function. We also know that there is a gamma secretase enzyme in the mitochondria. So this is that enzyme that second enzyme that cleaves APP. suggesting that you could, there could be a scenario where amyloid beta could be produced locally at the mitochondria. If we have an alpha or a beta secretase cleaving outside of the mitochondria, because to date, no one's identified that these are within the mitochondria. And so this is actually very interesting and important. There are some studies that have shown that the mitochondrial localized gamma secretase is important. If you knock it out, then the mitochondrial electron transport chain does not assemble correctly. And so this can cause, very bad downstream energy deficits in the cell. And if you even just, knocking out certain parts of gamma secretase, like the presenilins, in some studies does show that it can affect mitochondrial morphology and function. So it's important to note that APP, amyloid beta, they're likely in the mitochondria, and that they could be locally produced there. There are lots of studies showing, though, that mutant forms of amyloid precursor protein, which cause familial Alzheimer's disease, can also conversely induce mitochondrial dysfunction. And amyloid beta itself has been shown to cause mitochondrial dysfunction. So there is a strong relationship between amyloid precursor protein, amyloid beta, and mitochondria, as well as gamma secretase. So if you look at the literature, it can actually get very confusing, and it's very difficult in the Alzheimer's disease field because some studies have not been very, it's been hard to replicate them. But we do know that mitochondria affect Alzheimer's disease pathology in the form of amyloid beta production. And this has been shown in numerous papers. I've listed them all down here, starting in 1994, all the way to more current papers in 2012, 2013, and 2014. And I'm highlighting two papers here because they show slightly different results that I think are important to discuss and kind of fall into why I wanted to look at this. So in this particular animal model, this is an Alzheimer's disease transgenic mouse. So it's overexpressing human forms of amyloid precursor protein that are mutated in familial Alzheimer's disease. And these mice accumulate amyloid beta plaques in their brain, which you can see here as these dots. And what they did was they caused these mice to express a mitochondrial targeted enzyme which would cleave the mitochondrial DNA. So this is a nuclease. And so it's going to get rid of all of the mitochondrial DNA, and so that should cause mitochondrial dysfunction. And what's interesting is that getting rid of the mitochondrial DNA and inducing mitochondrial dysfunction, you can see there are way less amyloid plaques in the brain. And this is quantified down here and here in both the cortex and the hippocampus. And so that was very interesting. This paper actually concluded that mitochondria have nothing to do with Alzheimer's disease, and I think that this data means the opposite, that mitochondria have a lot to do with Alzheimer's disease because, messing with their function seems to affect amyloid beta production. In this paper, they crossed an APP transgenic mouse again with a different mutation. This is a mutation in polymerase gamma. Polymerase gamma is the proofreading enzyme in the mitochondria. So it proofreads the mitochondrial DNA to reduce the amount of mutations that accumulate over time, which can accumulate because of the natural reactive oxygen species that are produced at the electron transport chain. This mutation, though, renders this polymerase gamma non-functional. So these animals accumulate a lot more mitochondrial mutations over time. And what's interesting is that they saw the opposite. When mitochondrial DNA mutations accumulate, there's more amyloid beta being produced, and this is also quantified here. And so I think these papers highlight that there's obviously something going on with mitochondrial function to influence amyloid beta production, and we just don't really understand what that is. So I set out to try to figure out what that might be. And I'm going to steal my own thunder, and hopefully over the next few slides, I will convince you that mitochondrial membrane potential is an important factor in determining how much amyloid beta is produced by a cell. And so mitochondrial membrane potential is extremely important. You can kind of think of it as like an electrical potential across the membrane. If you lose this potential, you're no longer able to produce ATP because you're not able to pump protons across the membrane into the matrix to turn the ATP synthase enzyme and generate ATP. And generally, the matrix has an overall net negative charge, while the intermembrane space has an overall net positive charge. And this charge dissipation is what allows the flow of protons into the negative space, movement of this, I guess you could probably call it kind of like an engine, a rotor engine, to phosphorylate ADP into ATP. And we can measure mitochondrial membrane potential with really cool dyes. We use dyes that are cationic, so they have a positive charge, and they accumulate in the matrix when there's a negative charge. If there's not a negative charge in the matrix, then they won't accumulate there. And so we can really kind of look at if there's less red, then that negative charge is reduced. But if there's more red, then that negative charge is really high. And so we use, in our first phases of our studies, we've been using a neuroblastoma cell called SHSY5Y, so it's a neuronal-like cell. We use it because it's really easy to grow, really easy to manipulate genetically. And we can manipulate this cell line to where it actually doesn't have any mitochondrial DNA, similar to what was seen in that first paper with the animal models. And what's interesting is that the cells that don't have any mitochondrial DNA, which we call row 0 cells, have a significantly reduced mitochondrial membrane potential. And you can see that here based on the amount of red staining that we're seeing in between these two pictures. And these are normalized for cell number. So we know that we're not seeing less red because maybe there are less cells here than here. When we look at amyloid beta production, we see what that first paper showed. We see less amyloid beta 40 being produced, less amyloid beta 42 being produced as well. And the ratio is between those two is decreased. This ratio tells me about how the gamma secretase is functioning as well, because if we see differences in this, I can kind of indirectly conclude that there probably is some change to gamma secretase function in these cells. Which might make sense because these cells do not have a functional electron transport chain. And if gamma secretase is involved in forming the electron transport chain, it could be a compensatory mechanism for these cells to decrease their expression of this enzyme or use of the enzyme. So this is really interesting. Mitochondrial membrane potential depolarization, so reduction in mitochondrial membrane potential, seemed to reduce the amount of amyloid beta that was being produced. So I set out to look at other models of changes in mitochondrial membrane potential. And so we can increase or decrease the membrane potential using different drugs. And so one drug that we can use is called FCCP, and it's a ionophore, and it basically kind of punches holes in the membrane and just allows leakage and causes depolarization of the mitochondrial membrane potential, as you can see here. Oligomycin, on the other hand, actually inhibits A2P synthase, or complex 5, and by doing so, it increases the mitochondrial membrane potential because you're no longer able to pump those protons in there, so that negative charge increases in the matrix. What we saw with FCCP was in line with what we saw with our row 0 cells. When we depolarize the mitochondrial membrane potential, we essentially significantly reduce the amount of amyloid beta that is produced, both amyloid beta 40 and 42 in that ratio. Conversely, when we hyperpolarize the mitochondrial membrane potential, we see an increase specifically in amyloid beta 42. That was much higher than amyloid beta 40. I wanted to look at another model of hyperpolarization, and so one of the ways that we can also hyperpolarize the mitochondrial membrane is to starve the cells. And what this does is it does induce autophagy, and at shorter periods of time will induce mitochondrial hyperpolarization because they don't have the nutrients they need to make energy. And so you can see that starvation significantly increased the mitochondrial membrane potential, and as I saw with oligomycin, it did increase both amyloid beta 40 and amyloid beta 42 levels. So far what hopefully I've convinced you of is that when we depolarize mitochondrial membrane potential, we see less amyloid beta. And when we hyperpolarize mitochondrial membrane potential, we see more amyloid beta. And this is likely a change in gamma secretase function. So it's kind of like a balancing act going on between amyloid beta and mitochondrial membrane potential. The next thing I wanted to ask was, can we correlate membrane potential with amyloid beta? And so in the same cells, we measured hyperpolarization or depolarization, so that membrane potential with our dye called TMRE, and then we collected the medium and measured amyloid beta. And we saw significant correlation between mitochondrial membrane potential and amyloid beta 42, as you can see here. We did not see great or really any significant correlation with amyloid beta 40. But the strongest correlation we saw was between when we ratioed amyloid beta 42 to 40 and mitochondrial membrane potential. And this is actually a biomarker that is used in the clinic for Alzheimer's disease. These ratios in plasma and cerebral spinal fluid, they actually tend to be reduced in cerebral spinal fluid of people who have Alzheimer's disease. So that's actually very interesting. So overall, I can say that mitochondrial membrane potential does affect amyloid beta production. And this may explain why in some models of altered mitochondrial function, we see increases or decreases, because we don't know, for example, how expressing that mutant Pol G, maybe that hyperpolarizes mitochondrial membrane potential. So overall, it could have different effects on membrane potential, and mitochondrial membrane potential is closely linked to reactive oxygen species production as well. So these are things that we need to keep in mind moving forward. So as I said, we know that amyloid precursor protein targets to mitochondria. My question is why? What's the point of having it there? Is it there under normal conditions? Does it have a function? Because we actually don't understand what the function of amyloid precursor protein is. We target it in therapies. We target amyloid beta in therapies. We target gamma secretase in therapies, and those have not really been positive. In some cases, in most cases, it's actually shown that we've made cognition worse by trying to get rid of amyloid beta or inhibiting gamma secretase. And so I think we really need to take a step back and try to understand what is the function of these proteins before we start targeting them in human clinical trials. And so as it's shown here, amyloid precursor protein localizing to the mitochondria, as I stated before, there is a gamma secretase in the mitochondria that may locally generate amyloid beta. And there are enzymes related to this. This is called amyloid beta amyloid beta binding alcohol dehydrogenase, which could cause increased reactive oxygen species. There is insulin degrading enzyme in the mitochondria, which has been shown to degrade amyloid beta. And this is prion protein, which also enzyme which can also cleave amyloid beta. And so there are enzymes in the mitochondria that could be there to deal with proteostasis. And I know Dr. Ogbos told me that he'll be teaching proteostasis soon. And so when we talk about proteostasis, it's what we really mean is kind of a homeostasis of your protein. You don't want your proteins aggregating, you want them folded correctly. And when we talk about proteostasis, that's really what we're talking about. And so there were also some very interesting studies in yeast recently, which showed that mitochondria may act as kind of like this sinkhole for proteins that are aggregating. So that it could be a way for cells to get rid of proteins that are aggregating. They call it MAGIC, which I think is really interesting. And MAGIC stands for mitochondria as guardians in the cytoplasm. And so mitochondria are probably very important in proteostasis. And so to me, it makes sense that, that's why they probably, their function probably has a lot to do with how much amyloid beta is produced. And in addition to that, it seems like their function has a lot to do with how much amyloid precursor protein is at the mitochondria. And so similar to what I saw with amyloid beta production, when we measured how much amyloid precursor protein was localized to the mitochondria, it correlated really well with mitochondrial membrane potential. When we saw mitochondrial membrane potential was depolarized, so with the row 0 cell model or our FCCP drug, we saw more amyloid precursor protein at the mitochondria and less amyloid beta being produced. And we could block this with chloroquine, which chloroquine is an autophagy inhibitor. And so this makes me wonder if we aren't seeing some effects of amyloid precursor protein on mitophagy. And then opposite, if we hyperpolarize the mitochondrial membrane potential, we saw less amyloid precursor protein localizing to the mitochondria. So that's with oligomycin or starvation. And this could be reversed with chloroquine again, which is inhibiting autophagy. So there was a paper a while back in 2003 which mutated 3 residues on amyloid precursor protein, these 3 positive amino acid residues, and arginine, histidine, and lysine, 2 neutral amino acids. And mutating these 3 amino acids seemed to prevent it from localizing to the mitochondria. So we made this mutant, again, because we reached out to the author of that paper and they no longer had the plasmid available. So we made it again. And as you can see in our cells, based on how much plasmid that we express in our cells versus how much mitochondrial amyloid precursor protein we observe, in our wild type, we see an increase, a linear increase with the more plasmid that we express. But in our mutant, there's really no change in the amount of mitochondrial amyloid precursor protein that we're able to see. And those levels are actually pretty low, which you can see in our Western blotting here. So this is amyloid precursor protein, mitochondria that are more purified. And you can see a smaller amount here in our mutant than our wild type. And these are just showing purity. So we have no nuclear markers and really no cytosolic markers there. And this is our mitochondrial marker. And what's interesting is that this mutant may actually produce amyloid beta at a higher level than our wild type versions. And it also seems like it affects the amount of mitochondrial DNA that cells have. So when we express wild type APP and induce mitophagy, there seems to be less mitochondrial DNA as we would expect. And we see the same with this mutant version of amyloid precursor protein. This mutant actually causes a familial form. But this mutant, which of APP, which does not localize to the mitochondria, there doesn't seem to be any mitophagy going on when we induce it. And so this data makes me think that there is a role for amyloid precursor protein in mitophagy, and that's what our ongoing studies are looking at right now. So we need to address a lot of questions. Why does APP localize to mitochondria? And same question for gamma secretase. What are their functions there? Is the main goal of this interaction between mitochondria, amyloid precursor protein, and amyloid beta, is it to remove aggregating proteins? Does it have a role in mitophagy? And overall, what is the function of these proteins? Like I said, we still are not sure about that. So I'm going to take the last few minutes and just kind of quickly talk about some of the other models that we have going on in the lab. I've recently started, in the last couple of years, we've accumulated some induced pluripotent stem cells from non-demented and Alzheimer's disease subjects. And we can now generate from these stem cells, so these are what these induced pluripotent stem cells look like, neurons, which you can see here. This picture was actually sold through BioNexus a couple of years ago, auctioned off for charity. And we actually can measure mitochondrial function in these stem cell neurons. And we have a fair number. We have about 14 stem cell lines now as of this year. We have 6 non-demented, 4 sporadic Alzheimer's disease, and 2 familial Alzheimer's disease lines. And we can measure mitochondrial function in these cells. And what's really interesting, so this is oxygen consumption rate over time. And we put them through kind of a stress test, like you would do if you were to go to the doctor and you wanted to see how well your heart was working, they make you run as fast as you can. We do the same thing to the mitochondria, but we do that using drugs. And what's interesting is that we're seeing differences between the familial Alzheimer's disease and the sporadic Alzheimer's disease lines in the neurons. There might be differences here. I'm unsure at this point. This down here, we inject oligomycin, which, like I said, inhibits ATP synthase, and it tells us how much respiration is due to ATP production, and there doesn't seem to be a difference here. And these two are FCCP. So again, that uncouples the mitochondria and it makes them respire very quickly. And so there might be some differences here as well. And this is non-mitochondrial respiration. We completely shut down the mitochondrial respiration chain, and there seems to be no difference here. But what was also interesting is when we looked at glycolysis, so this is extracellular acidification rate, and this is a measure of how much glycolysis is going on, so how much glucose utilization is happening. As we kind of see in neuroimaging studies, the sporadic Alzheimer's disease subjects had less glycolysis than the non-demented going on. Well, this is non-glycolysis acidification, but then when we inject glucose, there's more actually here, which I guess is opposite from what we see in imaging. But if we're seeing less mitochondrial function, they might need to use more glucose. And there seems to be more spare capacity as well. And this is just shutting down glycolysis again here. So it's really interesting, this kind of flux difference in energy metabolism in these cells and these neurons. And so we need to continue looking at this. We also do see that these cells do increase amyloid beta, especially with how much time they're in culture. And so we see the Alzheimer's disease subjects are significantly increasing their amyloid beta production. We also can see some age-related differences. The blue is non-demented, but the black is young, so there's a significant age gap between these two as well. The other thing we've been doing is making cerebral organoids, which are also known as little mini brains in a dish. And this is kind of what they look like in the cell culture dish. Here we have data showing that they are expressing the expected neuronal markers, including synapses, immature and more mature neurons, markers here, choline acetyltransferase, showing that we have cholinergic neurons. And here, they're actually, the red is showing amyloid in here, and the green is a neuronal marker. Green here is the neuronal marker as well, and kind of the purple color here is actually an astrocytic marker, GFAP. So they do have neurons and astrocytes. The inner part of them, though, is necrotic. So that's one downside to using organoids. They also don't seem to generate microglia. So we've been generating microglia, and which you can see here in the imaging. These are microglia markers, CD11B and P2Y12. And we can actually put the microglia and co-culture them with the organoids to kind of make a more complete kind of brain system. And so this is something we've been looking at and using to test potential therapeutics as well. And what's also cool is that we can measure changes in amyloid beta production between different cell lines. We can also see that they phosphorylate tau, and they have oligomeric amyloids as well, and soluble APP alpha. So it's really cool because we can generate these models to really look at not only can we change mitochondrial function in them, but also how does that affect Alzheimer's disease pathological hallmarks. So from there, I'd like to say thank you especially to Dr. Swerdlow. He was my postdoc mentor, and this is my son. This was last summer when we could all hang out together without masks. We really missed that. We were at a little picnic, and he wanted to hold my child, and my child fell asleep. And so I told him that anytime I needed sleep, he was going to have to babysit him. He didn't agree to that. I'd like to thank my senior research associate, Sharon Manley. She's pretty amazing, especially during this year in uncertain times. And my funding historically has been through CAMBRI, the Kansas Idea Network for Biomedical Research Excellence, the KU Alzheimer's Disease Center, Landon Center for Aging, and the Neurology Department here, and of course the National Institutes of Aging, my current R00 funding. And then I'll leave you with cute pictures of my toddler and my dog, because everyone loves pictures of babies, and take any questions that you might have. So thank you.
Any questions?
Everyone's asleep.
If you have a question.
Feel free to ask me about anything, even if it's career related.
I'm going to go first.
Okay.
One of the more significant discoveries I've, as a non-Alzheimer's researcher, noticed in the last 5 years would be that amyloid beta undergoes circadian cycling.
Yeah.
And so is that all forms of amyloid beta or just A beta 42? And is there a difference between demented and non-demented patients with respect to circadian cycling?
Yeah, so it is really well known. So Alzheimer's disease subjects, they don't sleep very well. And we know that when you're sleeping, that's kind of when your amyloid beta levels reduce in your brain. So there's amyloid beta in your brain. Even if you don't have dementia, it's there. It's there at low levels though. So it's a normal part of physiology. We just don't understand what that physiology is completely. So that's important to know. It's when you get elevated levels, that's when people tend to get concerned. But yeah, so when you're sleeping, that's when your brain typically tends to get rid of more of the amyloid beta. And it's pretty well known that, like I said, Alzheimer's disease subjects, they don't sleep very well. That's a problem. And so it does make you wonder, do those sleep disturbances affect their amyloid beta load? Do the sleep disturbances start prior to the dementia and maybe they just weren't so bad so they weren't reported? And as to what forms of amyloid beta, I'd probably guess as far as I know, it's probably amyloid beta 42 that's only been looked at. That's the most popular species to be studied currently. That doesn't mean that we shouldn't be studying other species, but yeah.
Sleep is important, students.
Yes. Sleep, diet, and exercise. Those are the things that our center talks about the most with the general public. So, and staying active.
Baki, go ahead.
Well, thank you very much, Dr. Wilkins, and I really appreciate it. I didn't fall asleep because it's a very interesting subject to me. Yeah, you know that this is if I'm not sleeping during the seminar, this is a darn good seminar. If I'm fall asleep,
thank you.
all right, then I think quality is not that. Oh, okay, I shouldn't say anything. All right, Dr. Wilkins, you were mentioning about the autophagy and the mitophagy during your talk. I have seen a paper published in the Frontiers in Neuroscience. And this group, I think they're working at Northwest University. They discovered a mitochondria has its own mitophagy. In other words, the mitochondria eat itself. It doesn't go regular autophagy. Could you please comment on that one?
Yeah, actually, I'm very interested in kind of, I don't want to call them weird, we'll say novel and interesting ways that mitochondria are disposed of. I actually just submitted an R01 to look at some of these. I don't know much about mitochondrial kind of phagocytosing themselves, but I think that's fascinating, and I'll have to look into that. One of the things I'm actually really interested in is the fact that cells seem to, eject mitochondria. So in the brain in about, I think it was 2014, 2015, there was a paper that described transcellular mitophagy, where neurons would actually release mitochondria, and those mitochondria could then be phagocytosed by surrounding glial cells. And to me, it makes sense energy-wise because neurons can be very big, and so I think it's more energetically favorable to release the mitochondria at the synapse than to try to bring it all the way back up to the cell body, where the autophagy, mitophagy machinery probably are more, you know, found more in the cell body than they are in the dendrites. And then there were other papers that came out showing that astrocytes actually can give mitochondria to neurons, and that this may help with stroke recovery during the infarct to try to overcome that hypoxia. And that if you block this process, that stroke is actually becomes worse. And so I think that kind of these novel mechanisms of how mitochondria are moved around a cell, moved between cells, and phagocytose, whether it's they are doing it to themselves or other parts of the cell or even other types of cells, is very interesting and a really novel area that we need to study more. So
yeah. I was just carefully looking at their electron micrograph, what they were demonstrating, that the cristae shrinking, and then direct evidence. Well, I couldn't convince myself that when the mitochondria is kind of, the getting, the sick, there is a mechanism, what's called the fusion, that the healthy mitochondria gobbles up the sick mitochondria to utilize the resources. But it was I don't know.
There's another process called I can't say this. Instead of apoptosis, it's mitoptosis. Yeah.
Okay.
And so that's also newer, and it's kind of where the mitochondria that sequester themselves near the nucleus, and then they're either taken care of that way or kind of pushed out of the cell, and it's a mechanism for the cell to try to avoid apoptosis or cell death. So I think there are probably a lot of pathways we don't know about yet. So this is why basic science research is really important.
Right. We need to work more.
Yeah.
Thank you.
Dr. Wolf.
Hi, thanks. I enjoyed your talk. I'll confess to having only gotten a little sleep last night, so I was actually sitting there nodding a few times. But
it's okay.
The thing that was interesting to me, based on what you were just saying, was that one of the more fascinating things I saw while I was at Creighton University, we had like this neuroscience imaging thing, and I saw a movie of where microglia were followed in the brain, and they were running around, and you were talking about the mitochondria there, and I was remarking to myself, I went, oh God, it looks like they're picking up the trash, that's like going from house to house and picking up the trash, and it would make sense that that would have to happen because you take the neurotransmitter back up, but that wasn't the only thing in the synaptic vesicles. There was junk that was being spit out there, and that would accumulate if you didn't pick it up. So I thought that was way cool, and so it would make sense that potentially you could have the mitochondria being spit out there and taken up as well. But my question actually related back to your mitochondria thing. Another really cool imaging thing that I saw had to do with how the endoplasmic reticulum inside of the cell just danced all over and would come in contact with the plasma membrane and stuff. And then I was involved in a study years ago where we were looking at the motility of cancer cells, and we found that you had to have fission and fusion, mitochondrial fission and fusion. The ability of that to occur influence the motility of the cancer cells. And so with that background, I was curious, do you have any ability to look at the amount of motility in your mitochondria, the level of fission, the level of fusion, whether or not you get the great big long, more I don't know what efficient, I guess, would be a word for mitochondria.
Super mitochondria, yeah.
Okay.
So I've recently started doing some electron microscopy so we can actually see the mitochondria, because that's really the gold standard for looking at protein localization at mitochondria. So we've done some immunogold labeling, and also to look for mitophagy, so we can identify, if mitophagy is happening at a higher rate or lower rate. We also, right before shutdown and the pandemic, we got a SPED microscope. Do you know what that is? The super resolution microscope. And so it's kind of a bridge between confocal microscopy and electron microscopy, where you can resolve the cristae of the mitochondria using immunofluorescence. And so you get some really good images of them. And I have samples ready to go, but I can't use the microscope right now because we haven't been trained and we can't be trained right now because of social distancing measures. So in the future, I will say I hope to have some really pretty cool pictures and movies that I can show. So yeah.
The other thing that crossed my mind had to do with the ability of when you got the fission and fusion going on, they're changing DNA all the time between one another. And so I was, you know, it struck me that that could also be contributing to what you were seeing is that you got cells lacking the right DNA.
Yeah.
Or mitochondria, I mean, lacking the right DNA.
Right. You could be accumulating more mutations in one versus the other. Yep. It's really impressive how dynamic cells are. Their organelles are not static. They're moving constantly.
Ishan B has a question.
Yeah.
Ishan, go ahead.
Hi. My question was related to actually in the very beginning when you said that in living people that we can diagnose Alzheimer's by taking like a section of someone's brain, obviously. So like the only way to do it, radioisotope.
Yeah.
Is it that someone goes to see this, like, does someone go to see the psychologist first to know that they have Alzheimer's, or do they see the behavioral and then go to the hospital? Like, which way does it happen first?
So usually people are seeing their primary care and someone has a concern about memory, and they might go see a primary neurologist, which we have a lot of those here, and those are pretty common. And they may say, you know, it's probably dementia, but they may have a more difficult time diagnosing what kind of dementia. So then they would go see somebody like my postdoc mentor, Dr. Swerdlow, who's kind of the tertiary person that you would see. So he's a specialized neurologist who can run the cognitive battery tests that need to be ran. So the mini mental state exam is one of them. Look at gait, order MRIs and PET imaging, and analyze those to kind of determine what's going on. If someone wanted to get a lumbar puncture, then they may take some cerebral spinal fluid and look at amyloid beta levels there, and do other, you know, blood tests to make sure there isn't a vitamin deficiency or some other, you know, dietary thing that may be causing some cognitive changes. But yeah, so there are primary neurologists who kind of see everyone, and if needed, that person can then be sent to a neurologist who specializes in memory. And we have a memory care clinic here where people are seen for that reason. And our the main goal is to provide a diagnosis. And so I would say they're correct about 80% of the time without actually seeing the brain. So.
Yeah, I didn't know. I was curious because I was like, if you can't tell, you know, by doing the biological removal of the piece of the brain, then you'd have to be able to figure it out through psychology, behavior first. And
behavior is a big part. Some other kinds of dementia, you'll have, you know, age of onset is a big clue. Also, behavior is a big clue. There's some types of dementia where you have more inappropriate behaviors than you would with Alzheimer's disease, for example. So, yeah.
A guest.
Thanks.
Guest has a question. Guest, could you identify yourself? Unmute, guest. You're muted.
I think it's me.
Yeah.
I don't know why it put me as a guest.
Because you're not logged in through your single sign-on.
Oh, but I'm signed in. I don't know why it's going on. Anyway, so my question is, you know, in your mitochondrial depolarization experiments and your model. Have you ever measured the ROS, and did you reverse that with any antioxidant? Did you do that?
Yeah,
Because
I
I'm
have.
supposing that ROS is the reason.
Yeah,
for that.
we did measure ROS, and it didn't correlate with amyloid beta as well as the mitochondrial membrane potential did. But I do think ROS does play a role to some extent. My concerns with measuring ROS is that it can be very difficult because there's so many different species of it. So we could be missing a clue because maybe we're measuring superoxide when really it's, you know, a reactive nitrogen species or hydrogen peroxide. And so it's really hard to, there's no one way to look at all species of ROS at the same time. And I wish that there was. But yeah, we've done a little bit of ROS measurement. And,
So
and it was
question,
superoxide, and that didn't, mitochondrial superoxide didn't seem to track as well with it as membrane potential did, but that doesn't mean it couldn't be another ROS species as well.
okay.
Yeah.
Dr. Abbas, go ahead.
I want to continue my question.
He has another question.
Sorry.
So your data showing, you know, when you did the OCR and the ECAR, and it looks like they have some changes in that glycolytic capacity, right? After you add glucose and then the so did you see you know, that tells you that there is the glucose metabolism is changing there more than actually really OCR driven OCR, right? So did you try to correlate that with some, you know, earlier study where they did FDG PET imaging with patients Alzheimer and they saw some increase in decrease in FDG?
Yeah, so just because I'm seeing an increase in glycolysis doesn't mean, so when we talk about the FDG PET, we don't actually know if the reason why we're seeing reductions in FDG PET, if it's because the brain isn't using glucose or if it's not taking it up as well, because it could be either or. And so I think we can kind of try to use some of these cell models to maybe make inferences about that, but we have to be careful because cell models are not perfect. We try to make them as physiologically relevant as we can, but they're not the same as a brain. But we might be able to do some studies in our organoids, which I think we'll need to be doing. So yeah, it just to put it into perspective, to make an organoid, it takes 3 months. So it's a long cell culture game. So
yeah, I do cancer organoids and it takes time.
Oh yeah, they're fun.
Okay, thank you.
Dr. Abbas, go ahead.
Yeah, well, thank you for taking my question. Just a follow-up to Dr. Ihab Sarso's question. ROS is certainly important and then it's difficult to measure. However, I've just seen a recent the paper that they, well, actually it's not the kind of product that they develop a special dye very specific for the superoxide. If the superoxide level are high, the color changes or something like that, we can really measure. Because the life of the superoxide, it's very short, the kind of super millisecond. But this dye is somehow the captures that and changes make it possible. The other thing is that for the Ishan, the student Ishan, I forgot the last name, apologize. Certainly getting the brain tissue is already, the confirming that you got ready, it's already too late. We are actually working on developing the early biomarker in the blood base. Heather is in the KU and then I'm in the KCU side. We have a lot of collaborative work to develop the kind of blood-based biomarkers. In our laboratory, we are working on a new players in the AD named the TDP43, is a TAR DNA RNA binding protein. It seems that a kind of potential the features for being a kind of reliable biomarker, and we'll see how the research will go. Definitely, we need to have a kind of blood-based biomarker. It's easy, less invasive, and then it just tells the story before the clinical manifestations. So then the physicians will not miss the treatment opportunities.
Yeah,
Thank
so
you.
there are also some clinical trials going on. We call the APEX, which is the Alzheimer's Prevention Exercise Study, where they would do PET scans, and if there were amyloid elevated, is what we call it, then we would put them into an exercise cohort to see if they could reduce the amyloid levels. I don't think the results of that study have come out yet, but they're kind of trying to use some of these imaging techniques to capture people that have elevated amyloid and see if we can intervene earlier. So.
Okay, there are no more raised hands, so I'll give a call out. If anybody has a question, go ahead and raise your hand. I'll call on you. Students? Dr. Ogbas, I think you should wrap things up. Go ahead.
Well, thank you very much, Dr. Wilkins, for your time and your wisdom, as well as your data to share with us. This is critical because in the next semester, I'm going to teach the students the protein folding. Mm-hmm. And then all the way down the forming of the amyloid. It's really timely. And then, the seminar, Science Friday talks, that students already know what is the mechanisms or proposed mechanisms of the amyloid beta formation and then consequences of the clinical application. I really wanted to definitely, we have a collaboration. We would like to continue to work on this issue, and then would like to hear you sometimes maybe the next year what is update from your side, because this really, I have a primary interest that certainly I'm sure that it's a part of the kind of the global issue, let alone the the US issue. We are aging, and then we are really very much prone to get those neurodegenerative diseases. I would like to also thank Dr. Jeff Stadinger for organizing these meetings, and I would like to also thank all the participants for your time. And then, well, have a wonderful weekend. Thank you.
Yeah, thank you, everyone.
Bye. That was excellent, Dr. Wilkins. You hit the sweet spot for sure.
Oh, good. Do I need to stay on and talk to some students, or was that the question and answer session?
No, I think you do. That's what I heard. Is that still
okay,
true?
Yeah. I wasn't sure.
I'll stop recording now and leave the room, but you guys go ahead. Dr. White, nice to see you. All right. Take care, everybody. I'll stop recording now.